Hello everybody. We're going to start Unit 2 discussing structures inside the cell. In Unit 1 we discussed how cells are the basic unit of life. All living things consist of at least one cell. Cells capture the properties or characteristics of living things. Cells are able to metabolize, they're able to reproduce, they're able to sense and respond to the environment. If you look at cells under the microscope, you notice that they're very diverse. And we've already looked at some cells this semester. For instance, we looked at onion cells, we looked at cheek epithelial cells, and cells are really diverse, and nearly all of them require a microscope to be seen. The light microscope was invented in the 1700s, and its magnification properties are limited by the properties of light. In the 1930s, the first electron microscopes were invented. This overcomes this limitation by using beams of electrons to illuminate the specimen instead of wavelengths of light. The microscope on the left is very similar to the one that we've used in lab. Ours, however, has two eyepieces. The one on the right is a dissecting microscope, which we're not going to be using in class, but can be used for reviewing 3D objects. Say if you wanted to look at like the structures of a pine cone, something that wouldn't fit on a slide under a light microscope. This figure just gives you an idea of the relative sizes of different types of cells and cellular components. And you can see what's visible with a naked eye. So obviously, an adult, female or male, ostrich egg, chicken egg, frog egg, you can even visualize a human egg. Then you can see where you would require a light microscope to be able to see bacteria, plant cells, animal cells. And if you wanted to go beyond that and look at actual components of cells, such as lipids or proteins, you would need an electron microscope. Some proteins can be viewed with, yes, you would need an electron microscope. But why are cells so small? Why can't cells just be bigger? Well, they need enough surface area large enough for the entry and exits of materials. If you look at the picture here, we have on the left, we have a volume of four little cubes together, which gives you a surface area of 8. If we elongate the structure, you still have the same volume, but the surface area increases to that of 10. And on the right, again, still the volume of 4, but the surface area increases to 16. So you need a large surface area to volume ratio. Just think of the cube on the left. And the center portion would kind of be a dead zone, and it would be really hard for required nutrients to be able to enter and enough weights to be able to be eliminated. We've previously discussed the cell theory, and the cell theory states that all living things, all organisms, consist of cells. All cells also come from pre-existing cells, which to us sounds pretty obvious, but at certain points in time, the theory of spontaneous generation was something that was widely accepted. That meant if you put together the right types of substances and molecules, appropriate temperature, that you could generate bacteria. We now know that to be false. We also covered this in Unit 1. The properties of two different types of cells, prokaryotic versus eukaryotic. And this differentiation is based on the organization of the genetic material and whether the cells have membrane-bound organelles. Organelles are special types of structures that have a specific function. The nucleus would be an example of an organelle. Prokaryotic cells are much simpler, and they lack a membrane-bound nucleus. They also don't have any organelles. Where eukaryotic cells have a nucleus and have membrane-bound organelles. But keep in mind that all types of cells, whether they're prokaryotic or eukaryotic, have a cell membrane. This is an image of kind of a generic cell, and what it does is it gives you an idea of some of the primary organelles and internal structures. And some of these we're going to go over, and we'll just kind of go over structures that are important for our discussion of genetics this unit. We don't really need to cover all of them. This image is showing a generalized structure of a prokaryotic cell. And you can see that it has DNA, as all living things do, but it's not found within a nucleus. The word nucleoid means kind of like resembling a nucleus, which means it's located in a similar area where you'd find it in a eukaryotic cell, but again, there's no membrane surrounding it. Okay, which of the following structures would not be seen in a prokaryotic cell? Yes, you would not see a nucleus. Let's look at some parts of eukaryotic cells. The first is the cell membrane or plasma membrane. And just a side note, remember, this is also found in prokaryotic organisms. All living cells have a cell membrane, also called a plasma membrane. And what it does is it marks the boundary between the inside of the cell and the outside of the cell. And in lab this week, you looked at the passage of molecules into and out of the cell by the processes of diffusion and osmosis. So the plasma membrane acts to regulate the passage of molecules into and out of a cell. Remember in lab that we mentioned that it's selectively permeable. But now we're going to look at the specific structure of a cell membrane. It consists of two layers of phospholipids. And what a phospholipid is, it's kind of really similar to a triglyceride, and that's the most common dietary type of fat. There is a glycerol molecule and two fatty acid chains and a phosphate group. And these two types of molecules have two unique chemical properties. Polar heads, polar molecules, are molecules that can interact with water. So the polar heads of the phospholipid, that's where the phosphate group is, faces into the watery medium. The watery medium is found inside as well as outside the cell. The nonpolar tails, face each other. Polar is also referred to as being hydrophilic, which means water-loving, where nonpolar is also known as being hydrophobic, water-fearing. Now, this might seem kind of complicated, but you see this all the time at home. If you mix olive oil and vinegar and you shake it together, it appears that it's separated. But if you put the salad dressing down on the counter, very slowly you'll see that the oil will separate from the vinegar, right? So nonpolar molecules will only interact with other nonpolar molecules, while polar molecules interact with other polar molecules. So the olive oil, which is nonpolar or fatty, right? These molecules are not really dissolved. They're just broken into tiny little droplets. And then if you let the salad dressing sit there, the little droplets coalesce or come together, and then you have the salad dressing separating into its two phases, right? So looking at the structure of the cell membrane, you have on the inside and you have the outside molecules that can interact with water, which makes sense because the fluid inside the cell and outside the cell is water-based. But now you have this layer in the middle where all the fatty molecules align together. I always think of this if you had two people like laying in bed, right? One had their one head towards the top of the bed, the other person had their head towards the bottom of the bed, and their feet were touching in the middle, right? And where your legs and feet are touching, that would be the fatty layer, or the nonpolar part. There are a lot of other molecules that are found within the phospholipid bilayer. Remember, that phospholipid bilayer is the base structure that makes up a cell membrane. But there's more molecules that are found in there. There's proteins, there's cholesterol, and these molecules may shift and may move as needed. So the cell membrane is often called a fluid because the molecules can move around, mosaic because there's a bunch of different types of molecules that are found in there, mottled. This gives you a better idea of what a cell membrane would look like. So you see the base of the structure, the phospholipid bilayer, but it's embedded with a bunch of different types of proteins. There's glycoproteins, this is a sugar and a protein that's hooked together. There's glycolipids, that's a sugar and a lipid that are combined together. You see protein channels, you see peripheral proteins, and we're just going to go through a few of these different types of molecules that are found within a cell membrane. Celiac disease is really a disease of the cell membrane. In some individuals, celiac disease is triggered by ingestion of gluten. Gluten is found in wheat, rye, and barley. In celiac disease, the immune system attacks the lining of the small intestine. This inflammation can cause cramping, diarrhea, lack of nutrient absorption, which in some cases can lead to malnutrition. So the immune system response is really crippling the cell membranes of your small intestine where nutrients are normally absorbed. There are a variety of different proteins that are found in cell membranes. Some proteins act as channel proteins. They form little tunnels for specific molecules. An example will be channel proteins for chloride ions. To me, it looks almost like a little slide. So it's a protein that opens up the space between the phospholipid bilayer that allows chloride ions to enter in. So what happens if a chloride ion channel protein malfunctions? Cystic fibrosis is the most common serious genetic disease in the United States. It's caused by a single gene mutation that causes a cell membrane chloride ion transport protein to malfunction. This has a variety of manifestations. It causes the cells in the lungs to produce a thick, sticky mucus that makes the individuals more likely to suffer different types of respiratory infections. It also can obstruct the pancreas and make the absorption of nutrients more difficult. There's some really good videos that I will post in Blackboard after this presentation. Receptor proteins. Receptor proteins allow signal molecules to bind and cause a cellular response. Sometimes the molecules are binding might be a hormone, such as insulin. Diabetes mellitus is a disease where an individual does not produce enough insulin, or the insulin that is produced is not effective in regulating their blood sugar levels. Insulin is a hormone that's produced by the pancreas. It binds to these specific receptor proteins on your cells. This allows for the uptake of glucose from your bloodstream. People that suffer from diabetes mellitus have elevated blood sugar levels, which has a variety of systemic effects. Sometimes the proteins that are found within the phospholipid bilayer are enzymes, and these enzymes can directly participate in metabolic reactions. An example of this is the enzyme lactase. Lactase is an enzyme that specifically breaks down lactose. Lactose is also known as milk sugar. If you have a lack of lactase, meaning that your cell membrane does not contain these enzymes, lactose is not digested. Undigested sugars reach the large intestine, where bacteria there can ferment the sugar. This produces diarrhea, gas, as well as bloating. There's some really interesting theories on this because humans were the only mammals that continue to drink milk after weaning. We're the only mammals that drink the milk of other mammals. So the theory is that initially infants produce lactase because they need to be able to digest lactose in their mother's milk. But at a certain age, after weaning, infants no longer need to produce lactase, and so the production starts to reduce. Then as humans evolved and we started domesticating different types of animals and making cheeses and other products from their milk, the ability to produce lactase extended into adulthood. Lactose intolerance is very common in elderly individuals, so just over the course of their lifetime, the amount of lactase that's produced starts to slowly diminish. Sometimes the proteins that are found within the phospholipid bilayer are recognition proteins. Recognition proteins enable our body to distinguish between our own cells and the cells of other organisms, and these other organisms may be pathogens. Recognition proteins used in this way are commonly called antigens. What must materials pass through to enter or leave a cell? Here's another video that I will link on Blackboard for you to watch. The nucleus is the area of a cell that stores the DNA, the genetic material. In eukaryotic organisms, the DNA is organized into genes, and each gene specifies the production of a particular protein. The nucleus has a double-layered membrane around the outside of it that's called the nuclear envelope. And there are some small holes called nuclear pores that permit the passage of molecules into and out of the nucleus. Ribosomes are organelles, and remember the word organelle means little organ, that carries out photosynthesis in the cytoplasm. Ribosomes look like a little snowman made of two subunits. Some ribosomes are found free in the cytoplasm, while many are attached to a specialized structure known as the endoplasmic reticulum. Endomembrane system. This is a system that consists of the nuclear envelope, the membrane of the endoplasmic reticulum, the Golgi apparatus, and the vesicles. The function of the endomembrane system is the production, modification, packaging, and distribution of proteins. The word vesicle just means little storage container, and so transport vesicles are able to carry molecules from one part of this system to another. In this diagram, you can see a transport vesicle that is transporting proteins from the endoplasmic reticulum to the Golgi apparatus. And you can see it's kind of continuous from the nuclear envelope as it moves outward into the plasma membrane. The endoplasmic reticulum, or sometimes just called the ER, is a complex system of membranous channels. It's physically connected to the outer membrane of the nuclear envelope. But what does it do? There are two types of endoplasmic reticulum. There is rough ER. This is studied with ribosomes. Just remember, ribosomes are where proteins are made. And so rough ER contains the ribosomes. It forms and transports vesicles going to the Golgi apparatus. Right? The proteins are made on the ribosomes, and then they're transported next to the Golgi apparatus. There's also smooth endoplasmic reticulum, known as smooth ER. This is continuous with the rough ER, but it doesn't contain any functions. It's not involved in protein synthesis. The function may vary depending on the type of cell. Often it's involved in the synthesis of lipids, and it can also be found or function in the detoxification of drugs. This shows the difference between the rough ER that has all the little pink ribosomes attached to the smooth ER. The Golgi apparatus consists of flattened sacs, and it's receiving vesicles that contain proteins from the endoplasmic reticulum. And what it does is it modifies these molecules. It sorts them, it repackages them. It's kind of like the Amazon warehouse for their new destination. And some of these vesicles are known specifically as lysosomes. Lysosomes are specialized vesicles that can digest molecules or portions of a cell. They might be digesting portions of the cell that are not functioning properly. And the idea is to try to recycle some of the cellular components. So lysosomes are vesicles that contain digestive enzymes, enzymes that are able to break down molecules. Lysosomes are also found in specialized types of white blood cells known as macrophages. Macrophages have the ability to engulf bacteria and debris and to destroy it. Lysosomes can also be involved in cellular death. Apoptosis means programmed cell death, and this is a normal process that occurs during growth and development. If you think of a tadpole, it has a tail, and as it develops into a frog, the tail needs to be lost. Apoptosis can trigger the removal of this tissue. During embryonic development, human fingers are webbed together. Apoptosis is the process which can enable the fingers to then become unwebbed. Sometimes this apoptosis is not complete, and people can be born with their fingers or toes still adhered together. This is known as syndactyly and occurs in about 1 in every 2,000 births. Normally, this webbing disappears at about 4 months of gestation. Let's look at a disease caused by abnormal lysosomal enzymes. An example is Tay-Sachs disease. This is caused by a single gene mutation that results in a malfunctioning lysosomal enzyme. Alright, don't expect you to remember the name of these compounds or enzymes. But in normal brain and spinal cord metabolism, there's an enzyme called hexosaminidase that breaks down fatty material. When this enzyme is produced and it's not able to function properly, there's accumulation of gangliosides. So these lysosomes, instead of breaking down this lipidy fatty material, it starts to fill up. Children appear normal to about the first year of life, but then as this material accumulates, it causes brain deterioration. Tay-Sachs disease is often fatal by age 3. You can see the picture at the bottom, the microscopic view of an affected brain. Those white spaces or openings are accumulation of these gangliosides, which shouldn't be present. This causes disruption and death of the normal neuron cells. A tadpole that's undergoing metamorphosis into a frog and losing the need for a tail would see abundant numbers of what organelle to help assist with the tail loss? Vacuoles are membranous sacs. They're a little bit larger than vesicles, but they also act as storage containers. In some protozoa, they can help the cell get rid of excess water. They can also function in digestion and storage. Here you can see the central vacuole found in image B of a plant, a vacuole to help get rid of excess water as shown in diagram A, and a storage vacuole in diagram C. Energy-related organelles. Chloroplasts are found in plants and some members of the Protists kingdom is the site of photosynthesis. Photosynthesis is where solar energy is used to synthesize carbohydrates. Remember, it's carbohydrate, it could be a sugar, it could be a starch, it could even be a lipid. This is how these organisms store energy. ATP, on the other hand, is usable energy. ATP is generated by the mitochondria. Mitochondria found in both plants and animals. Mitochondria is the area where carbohydrates, or again, could be amino acids, are part of a lipid molecule, are broken down, and the energy from the breakdown of these molecules is stored in a usable form. ATP, or adenosine triphosphate, is a usable form of energy. Both plants and animals need mitochondria too. Look at the word cytoskeleton. It really means like the skeleton of a cell. It's a network of interconnected protein filaments and tubules that extends from the nucleus to the plasma membrane. The cytoskeleton helps the cell maintain its shape. Cilia and flagella are both involved in movement. Cilia is much shorter than a flagella and are more numerous. An example of the human body where you would find cilia are the cells found in your trachea. These cells are also associated with mucus-secreting cells. So mucus is released, this traps dust and debris, and the cilia moves to sweep these substances out of your trachea so it doesn't enter down into your lungs. An example of the human body where you'd find flagella is on our sperm cells. Here the flagella are much longer than the cilia, and they are singular. The flagella allow the sperm to be motile. That completes the first part of Unit 2, and we'll review these materials in class.